Hello everyone and welcome to another Latis Scientific webinar. My name is Richard Lawson, I'm the Technical Manager at Latis Scientific, and this webinar is covering an introduction to some of the microbiological methods we offer in our laboratory. The first and simplest microbiological method is the total viable count. The standard yeast extract agar pour plate method grows aerobic heterotrophic bacteria at a variety of temperatures. It's used to assess trends in bacterial content in drinking water and related water types, but doesn't have any regulatory limit. If you're working with closed systems, cooling towers, or recreational waters, there are limits on the amounts allowed to be present, and they should trigger actions if they're breached. The actions are specified in the relevant guidelines, whether that's BSRI or PUTAC. It does have a few limitations, and the main ones are that it doesn't show bacteria that can only grow in the absence of oxygen, and it doesn't recover organisms which are stressed, whether by temperature or by biocides present in the water, very well. And this is why most hospital TVCs use different methods, including low nutrient agars with longer incubation times and some better recoveries. There are a few key limits for it. So in potable water, as I mentioned before, there aren't any limits. It's used for trend monitoring. In bottled water, the limits for 22 and 37 degree TVCs, and it's important to remember that this is only for samples taken within 12 hours of bottling. Beyond that, the regulation states no unusual increase, which will vary for each different type of bottled water. In processed waters, whether it's closed system or cooling towers, they're used for measuring the general control of microbiological growth, and you've got limits for the numbers present, as in swimming pools, where there are two limits. So as in cooling towers and swimming pools, the lower limit is a level at which you should review all of your results and see if there are trends emerging which suggests that microbiological control is not as good as it should be. And at the upper limit, it suggests that perhaps you have a problem and need to take some more immediate action. In hospital purified waters, so these will be waters used for instrument water disinfectors, endoscope reprocessing, preparation of dialysis fluid. The limits vary by application and by the method you use, because there are a couple of slightly different methods you'd use for it. Moving on, the most common indicator organisms of microbiological contamination, whether in potable water or recreational waters, are coliforms and E. coli. So total coliforms are defined as gram-negative, oxidase-negative, rod-shaped bacteria which will grow at 37 degrees, ferment lactose, or express the beta-galactosidase enzyme, and they're used as a general indication of microbiological quality in water because they're ubiquitous in the environment. So their contamination of water with this wouldn't necessarily mean that there's fecal contamination in the water. However, E. coli one species within coliforms, and they'll grow at higher temperature, 44 degrees. They produce indole from tryptophan and express beta-glucuronidase, and these are used as an indicator of fecal contamination. Limits for both in drinking water, swimming pool water, and related things such as spas and hydrotherapy pools is 0 CFU bundles, i.e. they shouldn't be present. Bottled water has a slightly lower limit of 0 per 250 mils, but again, you shouldn't find them present in bottled water, and that's not just for 12 hours after bottling, that's at any point during the lifetime of it until it's consumed. When you're talking about recreational waters like lakes and rivers, you have a limit of 1,000 CFU, and beaches have a limit of 500 CFU. And the status of excellent is awarded if you get less than half that result. Pseudomonas aeruginosa, Pseudomonas species, and Pseudomonas are closely related things. Pseudomonas species is the first one, and this is often used interchangeably with Pseudomonas because a lot of things which used to be called Pseudomonas for example, Pseudomonas cepacia or Pseudomonas picketi, have now been renamed to Burkholderia cepacia or Ralstonii picketi. And they're gram-negative, oxidase-positive rod-shaped bacteria, and they'll grow on Pseudomonas CFC agar. And their main use in analysis is as an indication of the likelihood of a sample to be biofilm forming, as they do form biofilms and they tend to be present when either just Pseudomonads are forming biofilms or Pseudomonads and other organisms, which will also tend to form biofilms, are present in a system. And their presence in closed systems is associated with microbially induced corrosion, although it hasn't been shown that they directly cause it themselves. It's possible that they by forming a biofilm, will provide a nice comfortable home for some bacteria, which can actually then directly initiate microbially induced corrosion. Pseudomonas aeruginosa is a separate Pseudomonas. It grows on, we use a different agar to isolate it, so Pseudomonas CN agar, which is similar to Pseudomonas CFC agar you'd use for Pseudomonads, but it's got a higher concentration of selective supplements. You can also use the IDEX pseudolert method for it. We have accreditation for both. And in the same way as E. coli is similar to Coliforms, it's similar to the other Pseudomonas species, but it does a couple of extra things. So it produces a pigment called pyocyanin, which gives colonies a distinctive green-blue color, and fluorescin, which means it fluoresces under UV light. If you're doing it via pseudolert, your confirmation is instant because you confirm it straight after incubation. Otherwise, you test it via checking the oxidase reaction and checking that it hydrolyzes casein so it clears cetramide milk agar. When you're doing analysis, particularly if you're doing analysis for both Pseudomonas aeruginosa and Pseudomonas species, you should expect that Pseudomonas aeruginosa would always be less than Pseudomonas species. Pseudomonas aeruginosa is most significant as an opportunist pathogen, which can enter the body through broken skin or can cause eye and ear infections. And this is why it's mostly tested for in swimming pool or spa pool settings and healthcare settings, just because it has extra opportunities in these settings to get into close contact with people and infect them, or it has a vulnerable population in healthcare settings where it can potentially cause some very dangerous infections. In both cases, it should ideally be absent, and either repeated low positives or individual high positive results should trigger remedial action. In bottled water, there's a limit for this, but there isn't a limit for it in normal drinking water. And in bottled water, the limit is that essentially it shouldn't be present in 250 ml of sample. If you're looking at processed water in closed systems, the limits for it for Pseudomonads rather than Pseudomonas aeruginosa are 5,000 CFU per 100 ml 7 days after pre-commissioning cleaning, or 10,000 practical completion. Legionella are another major organism which we do testing for. So including Legionella pneumophila, they're a slow-growing rod-shaped bacteria, and they need iron and the amino acid cysteine to grow. So they're grown on either BCYE, buffered charcoal yeast extract agar, or GVPC, which is glycine vancomycin polymyxin cyclohexamide agar, which is a terrible mouthful and is a long list of supplements which suppress the growth of non-target organisms, because Legionella, it being a slow-growing organism, can sometimes be overwhelmed by faster-growing organisms when you're culturing it. When you look at the plates with them growing on it, they have a distinctive appearance of like the ground glass surfaces in ground glass joints in labware in laboratory equipment. Once we've processed them, we read the plates at 3, 5, 7, and 10 days. Anything which looks like a Legionella is a presumptive positive. We then subculture it to GVPC and blood agar, and then identify it with a monoclonal antibody latex agglutination kit. It's only once you have an identified positive result that you get a report of an interim positive phoned and emailed through to you. Legionella limits will vary based on risk assessments and application, but typically a result of CFU per litre should trigger a review of your control measures, and 1,000 CFU per litre would trigger action in your water safety plan. As culture methods, we also offer molecular methods, the main one being qPCR, which is Quantitative Polymerase Chain reaction. And instead of culturing bacteria, which can be very slow, Legionella can take up to 10 days, and mycobacteria can take even longer, up to a month. It makes copies of their DNA using amplification primers to make copies of specific genes or portions of DNA unique to a specific organism. And then you use fluorescent probes to measure the concentration of DNA present, and this is used to calculate the number of copies of DNA and therefore the numbers of bacteria in your original sample. And it's most useful for slow-growing bacteria, but in theory could be applied to anything. It allows you to deliver a result in 24 hours, where otherwise you could deliver much high. Otherwise, it will take much longer to get a result. Another major organism, this is really applicable for closed systems, are SRBs and NRBs. SRBs are sulphate-reducing bacteria, which do directly cause microbial influence corrosion in closed systems by reducing sulphate to hydrogen sulphides in any areas where there's anaerobic conditions, such as those under biofilms. Anaerobic conditions are quite common in closed systems because the goal in them is to reduce dissolved oxygen to prevent corrosion. There are two different tests for it. There's a 5-day and a 21-day test, and the 5-day test is becoming less and less popular because, although it's quicker, it's less reliable, and to get the same level of confidence in your results as doing the 21-day test, you would need to do at least 7 replicates of it. This is discussed in some detail in the appendix to BSA 552. NRB stands for nitrite reducing bacteria. These will degrade nitrate and nitrite components of corrosion inhibitors, leading to inadequate corrosion inhibition and therefore potentially pitting corrosion. As part of their metabolism, some of them will also produce ammonia, which can also be important in other forms of corrosion, such as stress corrosion cracking. This obviously covers quite a lot of ground, and in terms of the references for more accurate things, I'm not going to read them all out, but these are the currently, as of May 2017, up-to-date references for all of the guidelines in here. I'd like to take the opportunity to thank you all for listening. And if you need any more information, you can contact Latis Scientific either via email, info@latisscientific.co.uk, or telephone, which is 0208 853 3900. If you speak to the customer liaison team, either they will be able to help you or they'll be able to forward you to myself or one of my colleagues who can. Thank you very much.